Our research presenter today is MSU Associate Professor Eran Andrechek from the Department of Physiology. And his primary research focus over the years has been breast cancer, but he's going to talk to us today about lung cancer. He's an expert in how cancer moves from one part of the body to others. And he comes to us after doing undergraduate work and his PhD at McMaster University in Hamilton, Ontario. He did his postdoctoral work at Duke University, and if folks are in the mind to tease him, his laboratory webpage indicates that he gained an appreciation for Duke basketball while he was there as a postdoc. And he has a particularly appropriate task before him in talking about our current MSU work. In other venues and other days this year, Michigan State University is celebrating the 50th anniversary of the discovery of the cisplatin anti-cancer drug. And so Eran is in part of a long and socially important tradition of fighting the scourge that is cancer.
Thank you. Thank you for the opportunity to present our work today. So I'd like to give you a snapshot of our work, but what I really hope I can convince you as sort of a takeaway point is how we've used big data, mouse models, and some basic science research to now hopefully impact how humans are actually treated and hopefully how we can start to expand lifespan. So as Paul said, I'm going to be talking to you about lung cancer today. On the graph on the left, what you can see are the number of deaths as a percentage due to cancer. On the top, it's lung cancer. So lung cancer causes almost 30% of cancer deaths in America. When I say lung cancer, I know you're immediately thinking smoking. That's simply not the only cause of lung cancer. You talk about firemen going into buildings, toxic smoke, lung cancer. Radon, lung cancer. All these environmental exposures, you're talking about lung cancer. The other thing that you can see on the graph I'm showing here, the second line down on the right, it says smoking status. You can see in orange, those are the smokers. In red, those are the people that have never smoked in their lives. They're still getting lung cancer. So there's a genetic cause. Each one of those ticks looking vertically down is an individual lung cancer patient. You can see I've circled EGFR, so 14% of lung cancer patients have an amplification or mutation in this gene called EGFR. So what I'd like to do is give you a little bit of a basic background in lung cancer and why it's important, and then I'm going to tell you about what we've done and how it fits. So if we start by focusing on EGFR, EGFR sits at the surface of the cell, so it's a protein embedded at the surface. When the cell receives a signal to grow, it will bind to another molecule. At that point, the red portion in here is an enzyme. It causes a modification to be made to the tail of that protein. When that modification is made, there are signals that will tell the cell it's time to grow, it's time to survive. This is a normal process, this is good. In cancer, though, that red portion, the enzyme, is either always on or there's too many copies of this. As a result, we have an unchecked, uncontrolled growth signal to that cell, resulting in cancer. So what can we do about this? Well, there are inhibitors to that red portion, the kinase domain, so the enzyme in the protein. So specifically, we can go in with a drug that will bind to that region. It will block those modifications from being made to the tail end of the protein, and as a consequence, we block the cell growth and cell survival. And if we look across, we have 3 generations of inhibitors. We're adding 3 and 2.5 years with the first and second generation of inhibitors. So now we've got a mechanism where we can go in and in a very targeted way, we have a treatment that will just hit this piece. Well, what happens normally? How do we shut this off normally? This is how we shut it off as a cancer therapy. There's one more gene that I'm going to tell you about, and then I'm going to tell you about what we're actually doing. So this other gene is PTP. It's essentially shutting off this growth driver. So the bluish purplish region in this is actually going to go ahead and remove those modifications in the tail. So now we don't get a growth and survival signal. So this is where our research comes in now. What we've done in our lab is to take drivers of cancer. And we take those drivers and we inject those into single-cell mouse embryos. That results in genetically engineered mice that heritably develop tumors. We've taken these tumors here, and I'm just showing a snapshot of a couple of them. And what we've done, you've heard of the Human Genome Project. Well, we've done the same thing, but at the level of these mouse tumors. So with a little bit of money from the National Cancer Institute and the Office of Vice President for Research and Graduate Studies, we've done the whole genome sequencing from these tumors. Now, we don't want to cure mouse tumors. We want to cure human tumors. So we're taking the mutations and the hits in the genes from that mouse work, and we've filtered it through human data. And I'd like to tell you just a little story about one of the most exciting findings that we've got from the mouse work. So you remember I told you about EGFR, this driver of cancer, and this PTP, the thing that shuts off the driver. One of the things that we found was 80% of our mouse model tumors have exactly the same mutation in this gene that shuts off the driver. So if we have this mutation in 80% of the tumors that shuts off the driver, we've essentially kinked that protein. It can't do its job. Now it can't remove those modifications, and we have unchecked growth and survival. We can actually see that in the lab. In the panel on the left across the top, you can see EGFR, and there's a band in each one. It just says that we have that protein in each and every one of the tumors that develop. But below that, the left two, the pEGFR, it's not present in a normal situation because our thing that normally takes away those things is present and functioning. But in the mutant case, when we no longer can remove those modifications, now we can see that there. In the bottom, we have an antibody that can actually recognize, so you see the yellow circles, if you will, up there? We have an antibody that will bind to those yellow circles. When it's present and functional, you can see a brown stain. So normally in a tumor, when you have a wild type PTP, so the thing that removes that yellow, when that's present, we don't see it. But in the bottom, when we have a mutation, we can see brown stain everywhere. So it's essentially saying we have active growth caused by this driver of cancer. If we go back to human cancer now, and so what I'm showing you here in this plot is each individual column is a human lung cancer patient. 14% of those have either amplifications in the red here or mutations in the green in this EGFR, in this driver gene. Those patients then are treated with personalized medicine, so not cisplatin, not just standard chemotherapy. They're treated with those targeted inhibitors, so we treat very specifically the molecule that's disrupted. When we look at the mutation that we uncovered, we can see the same mutation in 6% of these lung cancer patients. That translates to 11,000 people in the US that are dying each year with this mutation. Those people are simply treated with chemotherapy. Our hypothesis then is, since we have a mutation here that we can't normally shut this gene down, we should be able to give them standard treatment for the driver gene because we're not shutting down that driver gene. So we've tested that in our mouse model situation. In a normal situation, which is the blue bar, we're only killing about 30% of the cells. So we're looking at drug treatment here. After 48 hours, we assess how many cells are alive. We've got about 70% of the cells still alive because we're treating a driving gene. In red, you can see there's only 30% of cells left after 48 hours of treatment. So we are specifically and effectively killing these type of patient tumors with standard of care therapy. So where do we go next? So we've just submitted this for publication. It's up for review right now. We need funding to continue the work. We've submitted an R01 just a month back. And we're also working with MSU Technologies. You simply can't patent a sequence, so we're trying to be a little creative to get around that. But once this basic work is complete, this is something that can quickly and easily move into clinical trials because these drugs are already safe and approved for use in lung cancer patients. It's simply a matter of saying, these are the patients that need this therapy. In terms of acknowledgements, we've got funding sources across the bottom, but I'd simply like to highlight that this work has largely been completed by one outstanding graduate student in my laboratory, John Renhack. And with that, I can take any questions.
Can I ask on the patent, that question, tell me how does one, do you seek to protect, or what's that?
So you can't patent the identification of a sequence.
Okay.
You have to have something more than that. So what we're proposing and what we're working on is more of a test to say, well, we'd like to put the sequence together with a couple of other tests, pool that all together, and now that's something that you could protect and patent. And then you could go to patients that come into the clinic, give them that series of tests, and then say, yes, you're one of the people that would benefit from this theory, from this treatment. So you can get the first generation inhibitor to give you 2.5 years, the second generation for 3 years, the third generation for another 3 years of survival. So you start stacking these things on top of each other.
Thank you very
All
much.
right, thank you.
That was excellent.